Ever pondered about the workings of the DNA structure? Well, you're in for a treat today. Our DNA, or deoxyribonucleic acid, is a complex molecule that carries the genetic instructions used in the growth, development, functioning, and reproduction of all known living organisms and many viruses. DNA is often compared to a set of blueprints or a recipe or a code, since it contains the instructions needed to construct other components of cells. The structure of DNA is famously a double helix, much like a twisted ladder. This structure is composed of units called nucleotides. Each nucleotide, in turn, is composed of a deoxyribose sugar, a phosphate group, and a nitrogenous base. These bases are the rungs of the DNA ladder, connecting the two strands and making the double helix possible. Now that you have a basic understanding of what DNA is and its components, we will dive deeper into the structure of the DNA molecule. Nucleotides are the building blocks of DNA. But how do they contribute to the DNA structure? Well, a nucleotide is a little molecular package composed of three parts: a phosphate group, a deoxyribose sugar, and a nitrogenous base. These components come together to form the backbone of the DNA structure. Now let's talk about ATP, or adenosine triphosphate. ATP is essentially a nucleotide with not one, but three phosphate groups. What makes ATP so special? ATP is like the energy currency of the cell. When a cell needs to perform work, it breaks down ATP, releasing energy. So, we can say nucleotides aren't just the building blocks of DNA, they're also the building blocks of cellular energy. The connection between DNA, nucleotides, and ATP is a testament to the elegance and efficiency of life at the molecular level. With that knowledge of nucleotides and ATP, we can further explore the intricate structure of DNA. Polynucleotide chains and bonds play a vital role in the DNA structure. But what exactly are they? Well, let's delve into the world of DNA to find out. Polynucleotide chains are essentially long sequences of nucleotides, which are the building blocks of DNA. These chains form the backbone of the DNA structure, providing it with its distinctive double helix shape. The backbone itself is made of a repeating pattern of sugar and phosphate molecules, hence the term sugar-phosphate backbone. Now, what gives DNA its unique diversity is the arrangement of nitrogenous bases attached to this backbone. There are four types of these bases: adenine, guanine, cytosine, and thymine. Each base pairs with its specific partner, creating a rung on the DNA ladder. But how do these chains link together? The answer lies in the 3-5 phosphodiester bonds. These bonds connect the sugar of one nucleotide to the phosphate of the next, holding our DNA strands together. These chains and bonds are the links that bring the DNA structure together. The most common type of DNA is the B type. But what makes it unique? Well, let's dive into the structure. The B-type DNA is characterized by a distance of 2 nanometers between the two polynucleotide chains. That's roughly 100 millionth of a centimeter. And if we talk about the distance between the base pairs, it's only 0.34 nanometers. It's so tiny, yet so crucial. Now, onto the concept of DNA helical pitch. This is the vertical distance along the axis of the DNA helix for one complete turn of the helix. The helical pitch of B-type DNA is approximately 10 base pairs. Isn't it amazing how these minute details come together to form the structure of our DNA? To summarize, DNA is a complex structure made up of nucleotides forming ATP structures linked together by polynucleotide chains and bonds, with the most common type being B-type DNA. Understanding this structure is key to understanding the blueprint of life.